Hi. Well, welcome to the Institute and to Murdoch University. It's a sort of wet Perth or rainy winter day. Reminds me a lot of Seattle, actually. One of the ways all this got started was, just reminds me of a day like today. I was sitting at Royal Perth one day seeing patients, and I got back to my email, and I had an email from someone at Microsoft Research, and said, we're computer people and we're kind of interested in HIV vaccines. Would you be interested in working with us? And I sort of replied straight away saying, sure, interested to know what you're doing. And we started working together a little bit, and David invited me down to visit Microsoft in Seattle. So I started working, came along. He said, would you give a presentation, not too dissimilar to this? It was actually in a very small room, and there was only David and 2 or 3 other people. On the way in, David said, would you mind signing this disclaimer? We just like to record things and put them on the server because people are time poor, but are sort of interested in work we're doing. So I signed, and he asked me for an abstract, so I called it something like using the most powerful tools of our time to address the most important problems of our time. And Bill Gates just looks through all this stuff and saw my presentation that led to a $12.6 million grant, which was the start of all this. So this sort of rainy day like Seattle, David at the end decided he couldn't stand all this rain. He moved down to LA where he was from. He's a very amazing guy, David. Subsequently worked with him for many years. But it just sort of illustrates that it's not with any kind of like malice aforethought. It's not like with any grand scheme or plan. It's just like being open to people and just, doing stuff, following your passion. You just can never tell what's going to happen. So, just with that little, I don't know why that story came to mind. Just because of these sort of rainy days, it's very much like Seattle. I'm going to tell you about, we use HIV as a sort of, I know you're doing infectious diseases, you're thinking about that in part of your coursework, but I thought I would use the story of HIV, and I'll tell you a little bit about the background of HIV, and see if that helps you construct a framework about which you might think about everything at a much bigger level. So, my story was I was working here as a medical student, and around the time I was a medical student, AIDS was being described as a new entity. A gay plague, wasn't really understood what the cause of AIDS was. And I'll tell you a little bit of the story of that in a moment. And I was always going to do a different specialty, endocrinology, but I was kind of, when we started to see the first few cases, there wasn't a specialty for looking after HIV or AIDS. There's no infectious diseases in Perth as a specialty like it exists now. And what had happened was that as these cases were being described in the US, the head of immunology, Roger Dawkins here, reached out to the gay community and started to engage gay men in coming into the Royal Perth Immunology Clinic and monitoring those men's immune function every 3 months, looking at their immunity in their skin, what's called a delayed type hypersensitive skins testing, and looking at their CD4 counts, and found, as they were in the US, that some of these gay men were having low CD4 counts and they were falling over time. And so that was the basis of a cohort, and I decided to move from endocrinology into immunology and as a registrar, and we really had our hands full in this time just setting up services for AIDS as it was then, and then HIV once the cause of AIDS became clear thereafter. And so these clinical priorities changed over time, and as an immunologist I also started to learn about other diseases, allergy, autoimmunity, lupus. And what's really striking is just the incredible progress that has been seen in HIV. It's a huge, huge problem. But what was it that made me switch out of my career plan into immunology and want to respond to that need? What was it that made Bill Gates care about this, and how I ended up shoulder to shoulder with people like that was not with a clear plan, it was just doing the right thing. Everyone just wants to do the right thing. Everyone is trying to respond to the biggest problems of their time. And that's the message, really. It doesn't matter where we are. We're in the world's most isolated city as we are here. If you follow your passion and do what feels important, respond to needs locally or globally, then a lot of good things will tend to happen. So we had these different priorities. You know, initially, obviously, the main problem was finding drugs that inhibited the HIV virus. And then we did that successfully, and then we started to see side effects of some of those drugs. And we were involved in a lot of the research that was in response to new problems that occurred in our patients. So in terms of the compass of what should I study, what research should I do, it's pretty obvious. The patient was telling you know, are you crazy? You want me to take all these drugs? They're making my fat disappear, making me look terrible. You know, surely there's something you can do to figure out what's going on and overcome that problem. Or, as with one of the drugs we were using, abacavir, 1 in 20 people would get really sick within 1 or 2 weeks of starting the drugs, usually within 3 weeks. And we would have a card that we'd give the patient, and it would say, watch out for these severe symptoms in the first few weeks. If you get these symptoms, then stop the drug immediately, ring us up. If you don't have the symptoms in the first few weeks, then you're going to be okay. The patient said to me, Steve, in fact, Steve and Wendy Walker have told their story in the Women's Weekly, so I can share this with you openly. Wendy is a teacher, Steve's an electrician. Steve said to me, surely you can come up with a test to predict whether I'm going to be the 1 in 20. So the question came from the patient, and we discovered a genetic test that predicted who would and wouldn't get the reaction, and that's what's called personalized genetic medicine, and that was a major discovery. So the discovery was exciting enough, that was incredible, the sort of making the discovery here and so forth. What was really tough was the next 7 years of translating that test. We used it initially in Perth, but then getting the whole world to be able to understand and use this test and get it implemented the world over. That was an incredible journey that was a sort of international collaborative effort involving thousands of doctors, investigators, patients, drug companies, regulators, all sorts of people. And again, you know, takes you in to engage with the world. But the issue is it's solving the problem at hand that really creates all this other great stuff. It's not, what I'm trying to convey is, none of this was with some brilliant plan that I wanted to, you know, do this stuff. It just comes from responding to the obvious things. So the other thing I wanted to talk a little bit about and use HIV as a way of getting you to think about, you know, one thing I found kind of confusing when I was at school and going into university was all these different subjects you have. You go, you know, the bell rings, you go from one history class to a geography class to a, you know, math class, and it just doesn't seem connected in any way. You know, how does this fit together? You know, what's going on here? And you basically just, you know, forget what you've just been doing. You go to the next class and you're in a different discipline, and you don't really doesn't make sense how this all connects. And it never really made sense to me, to be honest, until I'd been through a lot of these journeys with HIV, and it all started to kind of fit into a bigger picture. And this is a sort of framework in which I now place information when I get it. Where does it fit in this hierarchy? So, you know, when I was at your stage, I was really good at maths, physics, chemistry. You know, and I had a place at Oxford to do physics. And I loved it. It all made sense. I could prove a theorem, everything was fitted beautifully. And biology was like a mess. You know, there was so many facts, and it didn't kind of gel, and it wasn't satisfying. But then when I went to first year university here with some really good human biology and zoology teachers, I started to learn about evolution. And you probably learned quite a bit about Darwin and survival of the fittest and natural selection and so forth. And you know, it started to answer questions for the first time for me about why. So generally the way we sort of approach things is you see some observation, which is what? And then you think about, well, why is that? Why did that happen? How did that arise? And there's two broad ways of figuring out why for me from biology. One is evolution, where we came from, how life on Earth started. You know, the evolution of prokaryotes, eukaryotes, vertebrates, primates, down to us. And that evolutionary history explains a lot. One of the really important concepts someone told me about, again, first year human biology, was this concept of ontogeny replicates phylogeny. Has anyone ever heard that term? Ontogeny replicates phylogeny. It sounds kind of fancy, but all it means is if you think about a single cell that we start off with, single cells forming, and then, you know, gamete and embryo and fetus and then us. If you look at those images of what going from a single cell through something like a tadpole to a fetus to a neonate to us as adults, then it kind of looks a little like the evolution from a single cell organism through to vertebrates and primates, etc. It's just saying that when you track what happens from a simple start through to a more complex organism, it recapitulates, looks similar to evolution. So you basically have two kind of automatic ways of asking the why question. And in biology, we constantly, when we're saying, why is it so? It's usually we resort to evolutionary arguments or arguments about ontogeny or embryology, if you like, or just how we physically came to look in this form to address that question. And then the final question is how, the mechanism. So for those of you good at engineering, math, chemistry, the exact how of how mechanistically this is going on is the most challenging and important question. When you have the how, when you've gone through that whole process and you really understand the mechanism, then it gives you opportunities to come up with solutions, what drug to use or how to fix the problem or whatever. And generally funding bodies, research funding bodies pay us for answering the how question. Okay, they don't pay you to look at big data and say what is the observation. In fact, you're almost forbidden from asking why. You do it because we need to do it to develop a model that we then examine in the how, and that's how we figure things out. But the point I want to make in this slide is, although I learned about evolution, and because Darwin, thinking about biology, was the person who thought about this concept of going from simplicity to complexity, and how amazing things evolve in a biological context, the evolutionary process is not restricted to biology. It was going on long before the start of life. If you think about the Big Bang Theory and the formation of planets and stars and so forth, and then how that generated chemical complexity, and then out of that chemical complexity, biological evolution could start to evolve. It's the whole principle of what's called emergent properties. Does anyone know what I mean by an emergent property? If we take sodium chloride as an example in chemistry, you might do a project, one person might do a project on sodium and find out everything there is to know about sodium, and another person would do a project on chloride and find out everything there is to know about chloride on its own. And nowhere in that knowledge would you be able to know that when you put those molecules together and you get sodium chloride, that you end up with saltiness. Saltiness is an emergent property of sodium chloride that doesn't exist in either sodium or chloride. Something new. It emerged out of the additional complexity of putting those two things together. It's the first sort of concept of emergent properties. And that's a really useful concept, because suddenly you say, well, where does new stuff just spring up from? It comes from emergent properties, and that can come out of any of these evolutionary systems. And why stop there? With biology, what do you see layered on top of that? Based on our biology, the way our brains are wired, is mostly our brains are wired from what were the things that improved our survival as hunter-gatherers. So not getting killed by each other, or being killed by another tribe, or be killed by an accident, or starving to death. Those very basic things were the things that drove the way our brains are wired. It's part of why we're wired to hunt out fat, sugar, and food chemicals. And because our brains are wired that way, if you have Hungry Jack's and KFC and so forth competing next to each other, the store that packs more fat and sugar and things that we have food-seeking behavior from is going to sell more product. And the commercial survival of the fittest is going to see certain bad foods emerge as successful. So you can understand what's happening in terms of commercial evolution by understanding biological evolution. And so what I'm trying to convey here is none of these disciplines are more or less important. Obviously, understanding cosmology is very, very important and helpful. This evolution, to understand how chemical evolution occurred and where that chemical complexity arose from. And understanding chemical complexity was very important to understanding biological complexity. They've been going on over much longer, slower time frames. And so there tends to be an order to this. It's not that chemistry is more important than history that's more important than, but understanding the time frames and what the things at the bottom of the pyramid are more likely to influence what comes next. And that's a kind of hierarchy of how these things relate to each other. So how did HIV might exemplify some of those principles? Well, we now know there were these two different types of primates in Africa. And primates, with all these different primate species, you know, we're the successful culmination of this battle, but they're competing with each other for resources and food in the particular niche they find themselves. And they each have their own bugs, their own pathogens. There was types of precursors of HIV are called SIV, simian immunodeficiency virus. And there were two different forms of simian immunodeficiency virus, and chimps actually hunted these two types of species, types of monkey. And in fact, what happened was the virus, the SIV from this one and this one, actually combined to form SIV, and that nearly wiped out chimps. It's a sort of principle of us and our bugs, and the bugs that we do co-adapt to. If they don't kill us, they make us stronger in a way. They become part of our defense against competing species. And in fact, human bushmeat hunters were hunting chimps, and the chimps did the same thing to us. Their SIV came into man and became HIV. So this is we know the sequence of SIV in chimps, CPZ, is almost identical to HIV-1. So we now know that actually bounded between these two rivers in Africa, there's a particular species of chimps that gave rise to HIV. And they came, this was in Cameroon, came down the river links and the roads and the railway. And in the early 1920s, big cities were starting to form in Africa. And Kinshasa was a place that was a colonial city, and as it got bigger, populations became concentrated. There was prostitution, there was sexually transmitted diseases, a large population, and this SIV coming down in the bushmeat. Also, some people were keeping pet chimps, and they were getting scratched by the chimps. There were probably 2 or 3 transmissions. There were probably only 2 or 3 actual transmissions from chimp into humans where it took hold. And then there was a single, there were teachers from Haiti who went to this part of the world to help teach. And one of them, there was a single transmission back to Haiti. And then those teachers took, there was a number of teachers that took the virus back to Haiti, but there was a single migration from Haiti to the US around 1969 that was, we now know, took the virus for the first time to the US. But it took a long time before it was realized that HIV had reached the US. So the first AIDS cases were reported in about 1981, and it took a long time before HIV was identified as the cause. Now, why did it take so long? Well, first of all, when someone acquires HIV, it takes years before their CD4 count falls to a level that they get AIDS. It's called a long latency period. And so it took a while to realize there was a problem. And then because it arises so much later, the second set of problems were that the laboratory techniques at the time couldn't detect HIV, the usual virology tests. Fortunately, there was already a drug that had been developed as an anti-cancer drug. It didn't work in cancer, but was already made and on the shelf, called AZT, that had some activity against HIV in a test tube. But it really wasn't until 1996 that we had the triple cocktails that made it possible to suppress the virus continuously. Why would one drug alone not work? When you have a virus that's just growing at huge amounts, of, 2 billion copies per day, then if you put one drug against it, the virus just laughs. It makes a mutation, position 215, and that's it. The virus is now resistant to that drug. So when there's a huge amount of evolution, then it just solves the problem. It's just like having a lot of money to spend at the casino. It's going to get resistant. So when I first started treating patients in the 80s with AZT, they used to have to wake up at 2 a.m. to take their tablet. Within 6 to 9 months, the drug would have worn off and it would only have a temporary benefit. And so most patients in those days were expected to die, and so it was absolute revolutionary for us in '96 when suddenly with triple cocktails, combining 3 different agents, the virus has to make too many mutations at once and it's cornered, and now the drugs work. So this again was a real day-to-day reminder of me of the power of evolution. You'd go to clinic and every day would sequence a virus, you'd be able to see these evolutionary changes on the sequence. When you started AZT, you'd see the mutations emerge and you would see the patient get sick again. So it's unusual for us as clinicians. We got to see evolution day to day, and we saw the devastating consequences when you've lost the battle. So 1981, long before you, I don't know, Ronald Reagan was president. He was a Republican president. There was a lot of high inflation, gas prices were through the roof. He was quite reactionary at the time. These patients started to emerge with pneumocystis, this unusual pneumonia in the lung associated with a low CD4 count and a large number of these very exotic bugs in the lung. Prior to that, we'd only ever seen this condition in patients that had been receiving a lot of immune suppression from drugs for their treatment of their cancer. So we knew that something was knocking off their immune system, but what was it? A friend of mine, Mike Gottlieb, who gave me these slides, was in Los Angeles and a dermatologist who started to see these cases and reported in 1981 these AIDS cases. And they started to see these lesions too, this Kaposi sarcoma. Again, this was an unusual malignancy that was previously only ever seen in Mediterranean men usually. There was a lot of speculation as to what was causing this. No one really knew. Then the disease started to be seen in hemophiliacs, infants, prisoners, people that had been given blood transfusion. And again, no one had a clue what was going on. There was a huge amount of anxiety, as you can imagine, in the gay population, particularly in New York and San Francisco and places that had large gay populations. And there was speculation as to what the cause was. But as you can see, if each one of these dots represents 30 cases, there was really a concentration just in a few places, and there really wasn't a good response to the epidemic initially. It was then realized that in Africa, what had been called slim disease was the same disease, that AIDS was already there in Africa. And the first public health intervention, which was to ban certain groups giving blood, didn't occur till 1983. You might think that's quite incredible. Between 81 and 83, nothing was done in terms of public health. The big breakthrough of the cause of HIV, there was a big battle between the French and the US groups, Montaigne and the French group versus Gallo and the US. In fact, the first report of HIV in human tissues was made from a patient, one of our patients at Royal Perth, electron microscopy of their lymph node, and we could see these viral particles. That was John Armstrong, a really outstanding electron microscopist here, who made that observation. So even though HIV/AIDS was much later to come here, we already were able to make major contributions in Perth way back then. You know, in those days, the response was really poor. Obviously, once we knew the viral cause, we could make an antibody test and start screening the blood so that we stopped infecting people with blood transfusions. We were very fortunate in Australia that we did really well. First of all, we had universal free access to medical care, which was a huge advantage. And there was an early bipartisan political support. In fact, you know, Neil Blewett, who was actually an undisclosed gay man, was health minister, which was very lucky. Chris Puplik on the liberal side of politics was also gay. In those days, people couldn't talk about it when they were gay, they didn't come out. And so, but nevertheless, as a society, we were more tolerant and open, and it allowed us to make a really, what we now know to have been the best response to HIV/AIDS of anywhere in the world. We had a national policy with ring-fence money for treatment and care, education, research, and as a result, Australia led the world. In contrast, you know, in the US, there was a lot of fear and resistance to really talking about this. It was a huge shock when Rock Hudson came out to declare that he had AIDS, and Elizabeth Taylor, who's a film star, was considered incredibly courageous to come out and support her friend who had AIDS and to start to raise funds for AIDS research. Against the tide of conservatism, this little boy, Ryan White, was sent home from school for having HIV. He'd acquired it from blood products, and it really tested the US community response. And Larry Kramer formed an activist group out of the gay community called ACT UP. And I can remember when I first started doing talks for the first time in the US, it wouldn't be unusual for ACT UP to come in and start yelling with placards and throwing stuff at you on the podium. It was pretty scary, but it definitely did get people's attention to really what was an emergency. You can see here, these few years later, each one of these dots of 30 people, the way the epidemic is growing in the US. And AZT was licensed extremely quickly, and this activist response was really important to see this as the most rapid approval of a drug ever seen. So there was a tussle, if you like, within society to try and respond the right way to this epidemic. And you could think about this now when you think about, you know, no matter what your personal view is of climate change, what we're seeing now is how do we respond as a society, as a world, to climate change? And if you reflect on how we responded to HIV/AIDS as a global challenge, you'll see a lot of parallels in the way we tend to make progress in a kind of stepwise, halting fashion sometimes. It took this was the first time, I mean, just think, AIDS had been defined in 1981. The first time the word AIDS came out of the president's mouth was 1987. We actually had an advertising campaign called the Grim Reaper campaign, going before your time, I imagine, but it was really scary. It was a Grim Reaper, and there was bowling balls and knocking over children, and it was really scary. And so it was actually, in PR terms or in marketing terms or in public education terms, a spectacular program of ads, but it was many years too late. Can you imagine why this had to be shut down immediately? Why do you think there was uproar at these ads and it had to be shut down? It scared lots of little girls, because there was this cute little girl that was being knocked down by the bowling ball of AIDS and HIV. Well, it stigmatized the gay community, it stigmatized the affected community, and the affected communities said, stop, you can't do this is alienating us more. In fact, years later, the same communities said, bring it back, we now have treatments that work, and young gay men are not taking precautions, we want to bring this campaign back. So it was a brilliant campaign, but ahead of its time in terms of when society was ready for it. So this was an interesting Australian experiment. ACT UP was the activist group that would, a lot of activism when it first starts is pretty harsh and pretty unsophisticated, pretty scary, but over time became more sophisticated, and we were helped enormously by partnering with groups to help inform the general public. And activism we saw when it arose in Africa was again a little unsophisticated and sometimes did more harm than good, but over time evolved to become a very important part of our response to any problem. You can see the epidemic continuing to take off, the number of cases just going up and up in the US. This was the first George Bush Sr., and this was Saddam Hussein, the first Gulf War, where in 1991, Magic Johnson, the basketball player, came out and declared his HIV, which again helped demystify and open. Of course, in these days, it was still a death sentence, so people were really shocked that this person that was so important in their lives was likely to die of HIV and AIDS. At the conventions in the US, the Republican and Democrat conventions, you've probably seen these go on recently. This is the first time that HIV/AIDS was even mentioned, more than 11 years after the epidemic. So you can see how slow it was to just get people talking about it, acknowledging it, accepting it. This denial, initial response to a crisis will often be denial, and it's not about me, it's some other group. Randy Schultz, who wrote The Band Played On, a very important activist, he died in 1994. A film called Philadelphia about a lawyer who gets fired for having HIV came out, and that was helpful to shape public opinion. A dentist in Florida who had HIV infected a huge number of people. That was a big scandal in that year. So people started to feel more threatened themselves. Greg Louganis, a world medal, gold medal diver from the US, declared his HIV. The protease inhibitors came out around that time. And as you can see, this lack of response has devastating consequences for the US. This is where the science cuts in. Our understanding of HIV improves. It means we have a good test for HIV, viral load, the drugs start to work, we start to make progress, and you can see the difference between 1 drug, 2 drugs, and now 3 drugs. The viral load is continuously suppressed, and we see the number of cases of AIDS just plummet in response to these new drugs. But in those days, this would be the number of tablets a patient would have to take every day. And we started to see this problem of fat wasting in our patients. So we started to work on this problem here in Perth. David Nolan did some fantastic work here. Our patients are very generous. They allowed us to do over 300 fat biopsies. And initially, the people that described it blamed the new protease inhibitors because they'd just come on the scene for this problem. But we said, no, it's actually the old drugs. It's the AZT and the D4T. And we actually did some really nice research here in Perth. Again, we're swimming against the tide, but we basically were able to show that the drugs were affecting the mitochondria. They're the powerhouse energy-producing component components of the adipocyte, the fat cells, and this was causing a gradual loss of fat on the arms, face, and legs, and a redistribution to the gut. And you know, this is a patient of mine. This is normal fat with a nuclei, thin rim of cytoplasm, big, huge lipid droplet. This is healthy fat. And then this is a normal mitochondrial DNA level, around 700. And within 7 months of stabilizing therapy, you can see this increase in mitochondria. They're changing color, they're losing all the mitochondrially encoded proteins. A whole lot of the cells have died. So really a very nasty toxicity in the fat. And we wouldn't have been able to figure this out had our patients not generously allowed us to take little pieces of their fat to do these studies. Here you can see the epidemic marches on in the US. Now we have George Bush Jr., another war with the same Saddam Hussein, more war. Now we're down to 2 tablets. There's a recognition that we're doing well in the developed world, but in Africa there's a real discordance with this huge unmet need of orphans in Africa, this devastating problem in the developing world. And you know, fortunately we develop more and more drugs very rapidly, and combinations of those drugs. As I mentioned to you at the outset, that issue with Steve mentioning, you know, can't you come up with a test for abacavir hypersensitivity? We did the study here in Perth. We looked at the first 200 patients exposed to abacavir, and you can see, I can tell you, one sunny day outside Royal Perth, my computer expert had merged the data. We had blindly assessed clinically the patients, and we'd done the genetics in another lab, and the computer guy had put the data together and done the analysis we asked for. And I remember on a beautiful day in Perth with one of our bright blue skies, out showing someone around and going through my mail, and I just turned over the sheet and I saw this result. I have to say, it was literally one of those Toyota moments. It's the first time I ever jumped when I saw data. So, you know, of the 18 people who we classified out of the 200 of having abacavir hypersensitivity, 14 of them had this one gene. And we, over the next years, it was a long, interesting journey, ultimately working with international collaborators and the drug company. We did a definitive randomized clinical trial and got the perfect result that those screened to have 57 screening, not a single case of abacavir hypersensitivity occurred. And as a result of that, guidelines finally changed. I've really simplified the story. There were actually 42 hurdles to jump over these 7 years to get to roll this out globally. But it was a huge international endeavor with many, many heroes in the story. And now abacavir 3TC, this is now part of probably the top selling combined tablet. It's now combined with dolutegravir. But every patient gets the B5701 test beforehand, and those who are positive have an alternate drug instead. And it's eliminated the side effects, so it shows that personalized genetic medicine really does work. So now down to 1 tablet a day, new president, same problems. And what I guess that reminds us of is that our brains are highly attuned to defending against each other and to organizing our Army, Air Force, Navy into really well aligned defenses. But as you've seen in the story, we weren't well prepared in terms of our wiring, in terms of how to respond to an epidemic that was devastating and taking out much larger number of our population. And so we understand, because of the way our brains work and how we, both individually and collectively, that we will always you know, be at risk of underestimating pathogens as compared to each other as threats. And it just will ever be thus. It's not as if our biology can change. And so culturally, we have to change. We have to be aware. We have to adapt and evolve. And culturally, we can move forward far faster than we can biologically. I think this is a really important message that I certainly didn't understand early on, and that is that when the power of all of this, the change that you saw affected, that with the ACT UP, the change was affected by young people who just saw something wrong and reacted. And it's that passion, that ability to move things forward culturally that affects the most change. You can see we did well in Australia because we culturally were in a better place. And the US finally did really well when they culturally got there. But all the things we complain about, that we text about, that we rail about, that we're passionate about, that we talk about in a coffee shop or a ref or just day-to-day life, every time you feel yourself getting passionate, the tone of your voice go up, that you're contributing to cultural evolution. And being engaged in that and affecting change, what I can tell you is that's the most important thing I ever accomplished, was contributing to cultural evolution. In the early days, when there was fear around AIDS, I'd occasionally have to go down to, say, Albany and give town hall talks and reassure the population that even though there were people with HIV and AIDS in their community, and even though we didn't have therapies that were going to keep people alive in those days, that we could care for these people. And they did a tremendous job of stepping up once they had the information. But that's really what counts. It's the cultural evolution that makes a difference, and it makes a difference now. You are making a difference now. What you think now matters. That's the important message. So you can predict a lot of human behavior from evolution, but there are new emergent properties, things like the latest Pokemon stuff, you know, things will pop up that are emergent that you couldn't have anticipated till they, by trial and error, popped up and they were successful. Obviously, though, a lot of those things recapitulate and you know, somehow tap into our evolution. But in terms of history, history is a great guide to human behavior. But sometimes things change, like agriculture, food security, globalization. Things change, and so the rules change. And that forever shifts the environment. But piecing these things together, you can start to say what can and can't be predicted, and what things you should and should care about. When you're sort of tackling something, this institute, when we were building the institute, we often go through this process, a SWOT analysis, which you may have heard about. And when we went through the process here, thinking about some of the challenges we cared about, we realized that the first thing you need to think about is values. What are your values? And in the presidential elections you're seeing going on now, people care about values because it helps you predict what each candidate may or may not do. A vision, where would we like to be? Where would you like this institute to be 5 years from now? Vision is something imaginary, it's aspirational. What would you like to imagine yourself? Where would you like to imagine yourself in 5 or 10 years' time? What's the vision for yourself? And you know, this is the vision that we came up with for this institute. But a mission is how you get to the vision. People often confuse visions and missions. A mission is like going to the moon. It's a particular mission. These are the particular missions that we picked up. Often, if you were, as a group, to come up with a vision, you could all agree on what you would like to do. That's easy to get agreement on. You'll all have a slightly different idea of what the best way to get there is. And it's good to have that debate. That's good. You know, I just sort of wanted to give you that framework as to you know, what we do, why we do it, and you know, what we enjoy doing, what we care about. And the other thing I wanted to leave you with is that science is not an individual pursuit. All good things in the world have come from working together collectively. And having a shared values and having shared vision and shared missions is really what allows you to tackle some of these most interesting, important problems of our time. So with that, I've said enough. You'll hopefully enjoy looking around the Institute today. We also have a laboratory at Vanderbilt, so we have very strong collaborations with Vanderbilt, and we exchange technology to and fro and have these, virtually everything you see here, there's a clone of at Vanderbilt. If you ever come to Nashville, I'd be very happy to show you around there. And hope you enjoy your time today. So with that, maybe I could just take any questions that you have. It's a lot to take in a short time. You guys you've all done quite a lot on infectious diseases. Do you have any questions about any of the other infectious diseases you've heard about or worried about?
Do you maybe tell the boys why the HIV virus in particular? There's often vaccines and everything. Mm-hmm. Why is HIV so difficult with regards to formation of vaccines, whereas you're using treatment to treat the symptoms,
right,
as opposed to treat the virus itself?
Yeah, so that's a great question. The HIV virus, as you can see, is one of the most diabolically adaptable viruses. Because of this huge mutation rate, it's error-prone replication. Every single position of the virus, it's 9,000 bases long, and every single position mutates every single day without treatment. So the amount of mistakes, any one of those that has an advantage, that particular virion will get ahead. So its trick to survival is this incredible adaptability. And in general, the bugs that we don't have vaccines for are often very adaptable viruses. The ones that don't change, that everyone has the same variant, you can just mush it up, make it safe, inject it, and you have a vaccine mostly. So for a variety of reasons, the ones that are easy have just been done just by trial and error without really having to understand them. In fact, there is the Human Vaccine Project now, and I'm lucky enough to be involved in that project. It's a global project that industry and governments and universities around the world have come together to try and tackle not just HIV, but the other important pathogens, malaria, TB, and others, for which we don't have a vaccine. And there are fundamental scientific stumbling blocks. So really what's being done is to call time out on everyone working in little domains. So let's try and make an HIV vaccine here and a TB vaccine here and a malaria vaccine here. And let's come together and do studies that will address the fundamental scientific reasons that we're getting blocked. So that's one very major initiative in response to that exact problem. Basically, when you get stuck, most of what we have vaccines to, it's the old empirical trial and error, and we've got the vaccines for which that works, and the ones where we actually need to understand, we need to do some science. As you saw in HIV, we really couldn't make the progress till '96 until we'd done the science, understood the biology, and then we had the tests and the tools to address it. And that's really what's happening in HIV, a huge focus now on vaccine and also cure research to try and to actually try and eliminate the virus from people that have it, not just have them have to take tablets every day. So that's where the science really matters, and that's where hopefully some of you in this room might like to get engaged and help us with that. Yeah.
How do you define a zoonotic disease? Because I know some of us have been a bit confused about whether it's specifically just between vertebrates and humans, or if it could just be between any two vertebrates.
Yeah, so, I mean, I'm a physician, so I'm mostly thinking of zoonotic diseases in terms of diseases where, an animal has a, that where there's the animal soil that, and there's a reservoir, and then there may be a vector like a mosquito that there's often a vertebrate and then a mosquito, and then mosquito bites us. So, for example, dengue. Many of you will have been to Bali. Some of you would have come back with dengue. That's another huge problem. And again, there are these fundamental stumbling blocks of getting a good vaccine. So that falls in that category. So, having heard what I've said, do you think it's an accident that it was a chimp, our closest primate relative, that was the source of the HIV virus that caused such a massive epidemic? It wasn't something a bit more distant from us evolutionarily? Do you think, can you kind of imagine why primates that are very close to us are more threatening than a mouse, say, in terms of the bugs adapting? This concept of us and our bugs, and the intriguing thing about thinking about bugs, the way our thinking is shifting, we've all got mitochondria, right? And mitochondria are like a bacterial prokaryote that's inside us. We couldn't survive without our mitochondria now, but it's a little bit of prokaryote inside us, which is a eukaryote. So we're kind of familiar with the kind of Faustian deals we do in evolution, where you incorporate something that was initially an invader, you adapt to it, initially you put up with it, and then it becomes indispensable to you. Well, we're thinking about some of these bugs in a sort of similar way. There's this is a bit controversial in my own thinking, to declare that up front, but some of the viruses that we're meant to get shortly after birth, many of the herpes viruses, that glandular fever and so forth. If you were born in Africa and got glandular fever, you wouldn't get glandular, you get EBV, as it's called, shortly after birth. And it's probably the reason why we kiss babies and shake hands and pass our bugs around, those bugs to which we're adapted to, they trigger an immune response that shapes our immunity. Naturally in Africa you would get helminth infections, and you'd have a lot of worm infections, and you'd make IgE to worm. But we don't get that. Instead, we make IgE to house dust mite and pollens, and we get hay fever and asthma and all sorts of allergies and autoimmune diseases. So you can see the way our evolution has shifted, has taken away many of our natural pathogens at the natural time, and we're substituting more modern day diseases as a result. So we're trying to understand all these and try and figure out what is the right way to vaccinate our kids, and what should we be doing. So it's not quite as simple as we kind of teach initially, that they're the baddies, which are the bugs, and us, and we're all, there's good and bad aspects, even to bugs that we think we want to avoid. We're part of this world. That's just part of the thing. Makes you a lot more of a greenie in a way. You feel much more connected to our natural environment. We're part of our natural environment. This is all really artificial, and we've created something for ourselves, which is nice. I mean, it's freed us of traditional, food insecurity threats. We've created an environment which is good, but it's tended to let us forget our roots of the natural world that we're actually part of.
I was curious with that now we're down to one pill a day for the people with HIV. What is the cost on that for the patients themselves, or is that?
So we've touched with this has been again, in WA, we've really been lucky, and we still, touch wood to this day, our patients don't pay anything for the tablets, and we think that's really important. It's a very good, we do it for the right reason. I think the message from this is, we stepped up in Australia and we just did things for the right reason, and it turned out to be the economic rationalist things to do. We saved so much money as a result of that initial national policy, but that wasn't why it was done. They did it for the right reason, and it turned out to save money. And we believe the same thing. Giving people their HIV drugs free and getting them tested and treated early and stopping them infecting others is actually vastly more cost effective than allowing what you saw went on in the US to occur.
So is it free in America, or is that
No,
just an Australian initiative?
it's really just in WA, and we're just on the last legs of stopping the patients having to make a copay. Because there is, for some patients, what you always see is that, you know, and it's impossible to make judgments, and you should never make judgments about people. But people stopping their tablets because they're a bit short of money this month, or just going without for a few weeks, is really bad news. And it just isn't cost effective to, you know. And there is this sort of equity argument, well, if I have to pay for my blood pressure tablets, why should this HIV patient get their drugs for free? I think generally for infectious diseases in WA, we've always argued there's infectious disease legislation that used to exist that we luckily were able to point to maintain this. But if you have open TB, you get your TB drugs free. Yeah, HIV. It's nothing to do with it being HIV or any particular group. It's to do with it being an infectious disease, that it's more rational to give drugs away for something that is a communicable disease that's a threat to the public. And that is the reason why we think it's smart policy. But, you know, one of these days we're going to eventually lose the battle, and some people are going to have to make some of a copay. But today, WA has been very fortunate. In Sydney, they've always paid a copay, and there are people there that do stop and start their drugs as a result, and then you see resistance, and then you use more drugs, and it doesn't save you money, because you end up, you know, adding extra drugs on, which is vastly more expensive than the copay. So again, it's just, you know, I say, you know, just do the right thing. It will save money. I mean, we've shown it time and time again. You don't have to overthink this. If it feels like doing the right thing, do it, and pretty soon you'll be able to show that you're saving money.
I guess, yeah, this concerning part is then at a global level, if it's not being rolled out freely in America and Africa, then how do you stem that?
Sorry, that's a very good point. I skipped over that part of the story. Of all those presidents, I don't know if you know them, the Bush Reagans, Bushes, Clintons, Obama. Who do you think was the president that was responsible for giving billions and billions of dollars and getting antiretrovirals into Africa and the underdeveloped world? Who would you have predicted was the one who was responsible for that? Any guesses? Do you think it was hands up those who think it was a Republican, hands up those who think it was a Democrat. Democrat? Who would think it's a Democrat? Okay. Who would think it was a Republican? Okay. And which Republican was it?
George W. Bush.
That's right, George W. Bush. Does that surprise you? So the guy that went back in and did another Gulf War, you know, to finish off his dad's work, he was the one who funded PEPFAR, saved millions and millions of lives. What they've described as the single most spectacular execution of soft power the world has ever known. He did it for the right reason, you know. His wife influenced him, but it was really an interesting backstory of everything he had to do to get that massive amount of money to done. But it was a Republican, you know. So it's sort of interesting, you know. You know, it's not as if we're bad people, you know. We will act in a certain way. It's in our brains. It's in our wiring. But the capacity of people to eventually do the right thing and to see the benefits of it in terms of what does the most amount of good can come from either side of politics. It's important that there is a political debate and these ideas are competed against each other, and it's important that you are passionate for one side or the other and participate in this process. But you may be surprised which side of politics sometimes comes up. Sometimes it's easier for the right to do things like that than it is for the left. Okay. Any other comment?
Yeah, in the 90s, many doctors observed that many homosexual men had black pigmentation on their skin.
Yeah.
What was the cause of it? Is it the drastic reduction in CD4 or some other reason?
Yeah, so remember that first slide of the Kaposi sarcoma lesions, those black lesions? Yeah, so in fact it's known that there was two different viruses, the Kaposi's virus, another herpes virus, human herpes virus 8, caused by the HIV. When you have those two things together, then those black cancerous lesions can appear, and they really were horrendous. It was really devastating disease when we saw it. Fortunately now with the antiretroviral treatment, if you treat the HIV and the CD4 goes up, then the Kaposi's gets better. So we don't see that anymore because of the therapy. But you're right, these bugs, they've all evolved, and if you don't take care of the HIV in the world, what we've seen is a huge increase in TB on the back of the immunosuppression caused by HIV. So it really doesn't matter where the problems arise, you have to respond to these pathogens, and you have to, and again, doing the right thing, caring about what happens in Africa and responding to that therapy and getting treatments out there. If we don't do that, these evolutionary missiles from the past are going to come and get us here. That's, that's not why we should do it because it's the right thing. Do the right thing as soon as you realize that there's an unmet need. Because if you don't, as you saw in the US, there'll be a price that you pay for being slow to respond or late to act. Okay, well I hope you enjoy your day today and have more questions. And if any of this stuff, I think I sent you it may be a little, some of it may be possible to follow, but I think I sent you a book chapter that I wrote that had some of this material, explanation, references. So if you'd like to dive deeper about that, or reach out to me with an email, or if any of you ever want to come and do work with us, research, any of you might be interested in spending time at Vanderbilt too. A number of us have cross appointments at Vanderbilt and here. So don't hesitate to contact us. We'd be very happy to welcome you or your friends. We now have this sort of connection into the US for any of you that want to work or travel there.
Fantastic.
Okay.
Thank you so
Thank
much
you.
for your time.
I think Abra and Mark, we're going to have a little break to recover from this, and then Abra and Mark can come down.
Alrighty, yep. Thank you. Boys and predators, toilets just out there if you need to go to the loo, and recess and things like that.